this, right? Because they all have to beat all at the same time. And so that's very much a spatial question, right? Because you need like all of these cells to kind of work together in order to actually get to these coordinated and regular heartbeats. And so this is really a question that you're asking at many different levels, right? So it's at the cellular level. First of all, we need to make these cells, these cardiomyocytes. And here in terms of data sets, you can ask yourselves, right, in our body, we have many different cell types, and they can be very different, right? You have muscle cells, you have neurons, you have skin cells, etc. And in all of them, you basically have the same DNA, right? Your genetic information is the same in each one of their cells, but you still have these very, very different expression profiles, very different functions for all these different cells. And so the question is even, how is that possible? And so here in terms of data sets, or one explanation how this is in fact possible is if you look at this 1D information, so this is the DNA inside of each one of our cells, it's in fact packed in different ways. Okay, so I have always the same 1D strand of information, but you pack it up in different ways. And so if in one, it's kind of like a book, right? And so the stuff that I don't need for one cell, I will just crumble it up and I cannot access it. The other stuff is kind of like open in this book, right? And you can read it and you can actually express these genes. And so by different kinds of packings of the 1D information, of the genetic information, you can actually make very different cell types. So this is like quite important here of like, you know, what actually characterizes a particular cell type is, you know, how this, and this is here the nucleus that you're seeing as a picture of it, how this 1D information is actually packed up, which is often measured by this image. And on the other side, you know, what is the expression profile of all these 20,000 genes? So that's just at the cellular level. How do we even make, or what characterizes these cell types that are actually beating? So those would be these cardiomyocytes. Then you can ask a next question at the tissue level. And here is where you know, Xiao Wang has built really, really nice heart organoids. Okay, so these are, you know, you don't want to do it and take always data from the body, right? You actually would like to see this kind of outside of the body and actually be able to get measurements over time and see like how these heart cells are really, how these heart cells are beating. And here they can actually build heart organoids. So where these heart organoids are really growing and the cells actually start beating together. Okay, so you have these little heart organoids where you really have many cardiomyocytes, and they're actually starting, when they're maturing, they're really starting to beat all together. And so that's quite amazing in terms of data sets where you actually get to measure now using her technology, not just the expression of every one of these cells, but actually you also have electrodes in it, so that you can get joint measurements of the electrophysiological output, so all of these beatings, plus in fact the expression profile. And now what is important, right? The beating can be taken over time, but once you get the expression profile, the organoid or the cells are all dead. Okay, so expression profile, you only get at one time point, whereas, you know, the electrophysiological signals, you can get at multiple times. So that's at the tissue level. And then you can go all the way to the organism level and you can start studying different kinds of diseases that have to do with, you know, coordinated heartbeats. Like, for example, atrial fibrillation, which we had on the title, where you can now look at, you know, genetic information of people, for example, in the UK Biobank and many of these diseases, and really try to link it all the way from genetics to different kinds of disease phenotypes. And so really the question is at every level, right? If you really want to understand one of these diseases, you really need to be able to go at all these different levels. And what is quite exciting if you're thinking about organoids is you can actually then you can build even person-specific organoids with a particular genetic profile. So you can take cells from a particular person with a particular genetic background and then actually build organoids that will resemble their heart and try to see, you know, what is actually the mechanism of why are these cells maybe not contracting at the same time.
How good are organoids in terms of approximating the real tissue?
Yeah, this is always a very good question, right? So what is actually quite exciting here is actually that you can, so for particular genotypes that are well known for atrial fibrillation, you actually get phenotypes that are not all cells contracting at the same time. So at least at a very high level, they look similar, but it's always a question of like, how good are these? And that's why, you know, you of course also always do mouse studies, etc. in addition to that as well. Yeah. But here it's, if you really want to study like how are these two functional output and expression output, how are they linked? I think for that organoid like this is actually good, because they do start meeting together. Okay, so at every one of these levels, the problem you have is somehow, and this is very good that Paulina spoke last week, right? At every one of these levels, you have a data integration problem. And you have actually very different data integration problems. So here, for example, at the first level, right, we need to somehow integrate the packing of the DNA, which is usually shown or measured via these kinds of images of the cell nucleus, where very dense areas means that the DNA is packed very densely, and very open areas are represented here where the DNA is not as densely packed. And so you want to connect this to expression vectors, so these 20,000 dimensional vectors. And the challenge here is that you can never measure them together, because actually getting an image means killing the cell, getting the expression means killing the cell. So you'll never be able to measure both in the same cell. So you need to somehow, from these unpaired data sets, you need to somehow be able to translate and integrate between them. So it's not like what Paulina had last time, right, where you actually have paired measurements, where you have an image and you have a text for the same image. And now you try to get better representations. Here you have a population of cells, you take out some, you image them, you take out some, you sequence them. It's the same population of cells, but they're unpaired. I don't know which cell goes with which cell over here. Okay, but you can think of it that there should still be enough information in order for you to actually integrate these, because in particular, right, so if everything looks like a Gaussian, and I just get a marginal over here and a marginal over here, well, you don't know how to integrate them, right? There is like all this rotation symmetry that you cannot align. However, right, you have many different cell types. So say you have, some, I don't know, immune cells and some skin cells and some muscle cells, etc. And you take out some skin cells, some muscle cells, some neurons, and over here as well, right, for the other modality. And you already know there are 3 clusters. So you should be able to align, at least find a common space where the 3 clusters are aligned. So you can start breaking symmetries by actually having, access to additional structure like that. And we'll talk a little bit about how to actually do this. So that's the problem at this level. Then you go to the next level, and now here it looks quite similar to what Paulina had, where we actually do have paired measurements, right? We have paired measurements of electrophysiology and expression. But it's super important that we actually take space into account, because that's exactly the difference of spatial data to this not spatial data. It's like we want to know how much more can you learn by actually adding in that I know that this cell is next to this cell and next to that cell. Right, so this is a spatial information that wasn't taken into account in Paulina's talk, which I think we need to, because that's exactly what is new here for this kind of data set. And then at the end, you have like biobank data again here. I think one of the nice things is that you do have often quite a lot of paired data, right? So for example, here, if we're looking at heart data or heart diseases, we would maybe have, MRIs and ECGs, etc. But there you always have like this problem, which actually Pauline also talked about that, for some people you have paired data, but then there is often these data modalities like ECGs, which is much cheaper to get, where you'll have much more data on people, whereas MRIs, not everyone is going to get, right? And so the question is, how can you do well when you have a whole lot more data of one modality and only a little bit of data of the other modality that is paired? You should be able to gain from that as well.
Maybe I missed that, but like this is the switch biobank, right? There's these patients which have the whatever ECG or MRIs.
Correct.
This is one population. The population on which organoids are done, it's a different population, correct?
Sure, yes.
So there is no pairing like from kind of cell level to more phenotypical.
No, so not on the same UK biobank. Although when they're building organoids, it does come with a particular genotype. So they did use a particular genotype here in order to actually make this.
So the genotype that was specified in the biobank?
No, in this case, no, it has nothing to do with the biobank. But you can actually now do it for a person-specific basis. You can build an organoid, but they're not paired. They're not paired in the UK biobank at all. Yes.
Repeat questions.
Oh yes, I will do that. So this was a question about how is the organoid linked to the biobank.
So I had a question about the DNA package images.
Yes.
So you know which like the chromosome and then parts of each chromosome are packed. Can you tell that in the image?
Yes, okay, so I'll repeat the question. So if we're looking at this particular image, inside the cell, we have many different chromosomes. So here, actually, the only labeling you get is for DNA. So you just get to see, this is DAPI stained images, which you usually have a whole lot. You only get to see, like, for the whole chromosome is, so I cannot link it at the chromosome level. You can, there are images like this as well, much harder to get. And then usually you have 3 colors. So you would measure like 3 different pairs of chromosomes. That data is also available. Yeah.
So the information you're getting out of the images is the general amount of the DNA.
So it's actually, yeah, so I'll go to this on the next slide. In fact, even the shape of the cell nucleus gives you a whole lot of information about, for example, what the cell type is, what the mechanical state of the cell is. That already contains, even pathologists actually, the staining of the cell nucleus is very, very informative in cancer states. And it's also, it has to do with also like how condensed is it? Where is the condensation? Is it like more at the outside, more at the inside? There are all kinds of patterns that actually matter in terms of the condensation of the DNA. This is a great question. And in fact, this is exactly what we try to do in this first one here. So this is at the cell level now. The question is, how can we integrate and translate, right, between these very different data modalities? So we have these expression data on one side. So these are these 20,000 dimensional vectors of how much is every gene expressed. And then on the other side, we have these kinds of images, these DAPI stained images. And this is this question, right, where we have a whole lot of cells. We take out some for imaging and some for sequencing. And now we kind of want to find an integrated representation. And we, in particular, would really like to be able to go from imaging data directly to expression, because it's much cheaper to get imaging data than it is to actually get expression data on each one of these cells. And so here, how we did it here, and you know, Paulina really nicely talked about these kinds of models. So we're here also using, and we have really come to love autoencoders in general. So we also use autoencoders, and we also have a latent space that integrates these different kinds of data modalities. And so she was using contrastive loss in the latent space, and we'll be using contrastive loss also afterwards when we have paired data, right? When you have paired data, you can use contrastive loss. You say like the ones that belong together, they should be closer than things that don't belong together. But here we don't know what belongs together. So the question is, how do we deal with that, right? And you know, what you do know though is that since, you know, I had a population of cells, I at random chose some out for imaging and some for sequencing. Well, the population of cells is the same. So in the latent space, whether I come from imaging land or from sequencing land, it should be the same distribution. So what I need to do is I need to find an embedding, right, where I cannot tell, if I give you a point, whether you came or you came from imaging land. So I'll just discriminate. I want the 2 distributions to be the same in the latent space, whether I came from imaging land or for sequencing land. So that's how we do it here, to actually find these, to find a joint embedding of images and sequences. And of course, once you have that, right, once you have an autoencoder, this can be done for any number of modalities. Once you have an autoencoder here, for example, that goes from imaging to this latent space back to images, and here from sequences to this latent space back to sequences, well, then what you can do is just take an image, right, the real image, say, map it to the latent space, and then take the decoder out to the expression space. And what you get out is a predicted or a generated expression profile for this particular image. And of course, you can do it the other way around, maybe not as interesting. We usually want to go from cheaper modality to more expensive modality. And in fact, we validated this. Now you can ask, well, you know, I said you cannot do that, right? Either you kill the cell, you get an image, you have to kill the cell, you get an expression profile, you have to kill the cell. So what we validated here is, in fact, using these different colors in an image, where we say like, okay, so let me just choose a couple of genes. Okay, and since we have like at least 3 color channels, we take 1 for DAPI, and then also 2 more for 2 proteins. So I just choose 2 genes, right? I know. So I'm going to go from this DAPI stain, I'm going to predict gene expression. I'm going to take 2 genes, and in the image, I'm also going to color for these particular genes. Now, of course, expression RNA-seq is not the same as proteins, but you choose some genes where it seems at least that RNA and protein are quite well correlated. And so then you can predict the level of these particular genes and at least validate it for 2 genes. Okay, we cannot validate it for 20,000 genes because we cannot color for all these 20,000 genes. But at least you can validate it for a couple of genes so that you can actually do this. So that's how we did this for integrating very different modalities, right? Something like where we didn't use that we know where each chromosome is. Otherwise, you could have like some kind of supervision, but we don't have anything like that. They're really completely different modalities. One is imaging, where you don't have anything where you know where each chromosome is and how it relates to genes. And on the other side, you have gene expression. But you just use the intrinsic variability of data inside the particular cell type. Okay, so now then we're moving up to spatial data. And so now here we have not only gene expression, and we haven't done it yet with the electrophysiological input, but what we do have always in spatial data is we have this gene expression data, right? And we also have always these DAPI staining that you saw here, the same images. You actually have that in the image as well. Okay, so you always get to see, at least in star map, you always get to see also these images here of where the nucleus is and the staining of it that we just had on the previous slide, where we integrated expression and the stain of this of the DNA packing. So those 2 you now have, right? But now it's paired. Now this is the amazing thing, that now in the tissue, I get expression and I get the DAPI staining. And now what we would like to do is say, like, well, how much more information do I get if I actually add in the information that this cell is next to this cell, and this cell is next to that cell, right? So you also have an adjacency matrix over all of the cells, because I know how they were in this tissue of which cell is close to which cell. And so now the question is, how much better does your latent space become, right? When you can actually use all of this information when you're getting your embedding. And so now in this case here, how we're doing it in the latent space, so like Paulina talked about last time, you could have a contrastive loss that would give you, that's not enforcing that every one of the pairs that they're the same are really together. That's just, encouraging the right pairs to be together. Or here what we did is just an exact match. So if you do mean squared error of these 2 embeddings that you get from images or from expression, that means that you're just enforcing like every pair to be close, to be exactly on top of every other pair, so that you get the same embedding. So that's how we did it here on this expression data set. You have other problems in terms of this data that's like highly zero inflated. So you usually don't just reconstruct the full expression data. You usually just reconstruct based on a model that you're trying to estimate. But just in order to show you how this works and how different it is when you're actually using information about who is close to whom, this is the kind of clustering that you're getting when you're doing clustering now in your embedding space. And this is again for this hippocampus in the brain. So this is a mouse brain. This is what you would standardly do if you don't add adjacency matrix. Okay, so this is just a fully connected network. Just your standard autoencoder. And you see that here, it's really using neighborhood information, right? It's finding clusters that are actually also close in spatial state because it's using the adjacency matrix. So it's actually finding new clusters that it wasn't able to find when you're just doing it based on, this would be the same as what you would get out for single cell RNA-seq on this side, where you don't use any spatial information at all. So you get new clusters, so you can ask, well, the clusters have to be meaningful, right? Otherwise, this is useless to do. In fact, and this is something that, Shimi is now looking at, right? So it seems like, for example, for Alzheimer, these clusters actually matter. So there seem to be clusters here that are ahead in the disease progression as compared to other clusters. So we have mouse data from 18 and 13 months in this Alzheimer's disease progression. And so it seems that, you have larger clock in one side, and already at eight months, you already look like the next area at 13 months. So it's quite nice that it actually seems to be biologically relevant, the kinds of new clusters that you find here when you're adding spatial information to it. So that's like kind of a different problem, right? Of not just clustering based on expression, but actually clustering based on expression and spatial neighborhood. So you want to find spatially clustered regions that are similar in terms of their expression. Okay, so that's at the tissue level. Now you can go one up and you can be like, well, now I have MRIs and ECGs, etc. And I'm at the patient level. And now I would like to find integrated representations of patients. So I would like to find better representations of patients by using, all of the different data modalities that we have on a patient. Now here, because we're doing it on the heart, we only use 2 modalities for now. And now this is really like what Paulina presented, right? Where we're actually taking very different modalities. These are quite complicated modalities. I mean, these MRIs are like actually 50 frames of images because it's taken over time. So it's really like a video. And here you have an ECG. And they're both embedded now again paired, right? So we can do a contrastive loss in the latent space where we're encouraging, images or data points from the same patient to be close and data from different patients to be far away from each other. So that you can do using a contrastive loss. And then, what are questions that you can ask? Well, you can do exactly, this is quite nice. So I just took the same image on the next slide that Paulina showed, where you can, try to predict a particular phenotype from, say, for example, from ECG, you could try to predict an MRI phenotype because ECG is so much cheaper than, for example, MRIs, right? So you can predict that. You can just translate directly, so go from one modality to another modality. Or, and this is, I think, something that we're really excited about, is, what people want to do quite often is to actually find the relationship between the genome, the genetics, right? Your SNPs in this case. So what is your genetic information and different disease state? And so if you have a better representation of a disease, you might be able to better find the variants that actually cause this particular disease. So you can have a better representation of a phenotype in the latent space. You might actually be able to do better genome-wide association studies in this latent space and actually find more variants that are really associated with a particular disease. So in terms of these latent spaces, this is exactly the plots that Paulina showed last time. So I thought we can just show them as well, right? So for example, if you have, you have ECG data and you look at an MRI-derived phenotype, and so the question is, well, if you have just one of the modalities, how well do you predict that particular phenotype? If you have 2 of the modalities, how well do you predict that particular phenotype? And exactly like in her plots, right? Also for these modalities, always if you have your joint latent spaces, you can do better than, if you would, in this case, for example, just have your ECGs. So that's really just the same as what she had shown, which is quite nice. I don't really have to explain that anymore. What I think is nice, and this is, the other question when you have like some a cheap modality or a cheaper modality to get where you have a whole lot of patients that have that, for example, here ECGs, and you have other modality like MRI, where you often have much fewer patients, then you can ask, well, is there something to be gained, right? So for getting the same kind of predictive value here of, when you have like a whole lot of MRI data and very few kind of ECGs, it in fact turns out to be about as good as having much less MRI data, but a whole lot more ECGs, right? And so you can ask in terms of this, it's quite nice that it actually looks kind of linear, right? You can ask, what is your trade-off? So if I have a couple less MRIs, which is super expensive to get, how many more ECGs would I need of other patients in order to actually get basically the same kinds of accuracies out? Right? This is like a quite important kind of curve to know, because this means that you then don't need a lot of paired data. So you don't need that much MRIs and ECGs. You might just need very few MRIs, and then it's maybe much better to just get a whole lot more ECGs of all kinds of other people, and you might actually be doing equally well. So that's these kinds of scaling laws, where it would be nice to prove something about them, but this here is just done, just experiment. Okay, so like in Paulina's talk, like I just wanted to do one theoretical kind of insight, and then move on to the discussion. And so here, all I want to say, and maybe I won't go through the theory down here, But what I want to just talk about very briefly is that, I'm sure that many of you, when you use your neural networks, you know this, right? That, we're far off. I mean, we already understand this quite well, that, there is often no such thing as overfitting, in the sense that, if you're making your models larger and larger, it's often the case that they actually do better and better, right? And so this has, theoretically, we have understood, and this is a lot by work by Misha Belkin, that there is this double descent, right? That, at some point, yes, you do see your standard overfitting curves, but then at some point in these neural networks, it actually often just comes back down that even though you're making your models larger and larger, they actually don't overfit, but they will actually perform better and better. And so we ask similar questions for autoencoders. So, autoencoders standardly, right, you have a bottleneck. So you put in, right, you have like some input that is like, I don't know, in this case, 20,000 dimensional, we go to 100 dimensions, and then we go back to 20,000 dimensions. But, if we are thinking in the same way as for classification networks, well, why not just go to a larger dimensional space, latent space, than what you started out with? Now, it's maybe a crazy idea, right? Because this means you can just learn the identity map. You have enough parameters to just learn the identity map. But what we showed is that that's actually not happening. Okay, so if you just do gradient descent, and from experiments also, but in the proofs, just gradient descent. In experiments, of course, also like things like stochastic gradient descent, the functions you're learning are in fact not the identity map. So you can make your space much higher dimensional than your input space. And we did show also what are the functions that are learned. And these functions are in fact contractive at your training examples. So they really don't look like the identity map. They are, in fact, if you just iterate your maps, so, you train your autoencoder and now you put in a new image, right? If it was the identity map, then something comes out that looks very similar to whatever you put in. That's, in fact, what an autoencoder does. But now you just iterate it. Whatever comes out, you put it back in. And you just iterate it. And then, that tells you a little bit about what the map is that the autoencoder learns. And you'll see that it actually converges to one of your training examples. Okay, so yes, whatever came out looked like the identity, right? It looked like whatever you put in as a test image, but it's not. Okay, because if you iterate it, you're actually going to end up with one of your training examples. So the map that is learned, even though you're over-parameterized, is in fact not the identity map. It is a very interesting kind of map that is contractive at the training examples. And in fact, what we showed is that what is actually learned is something that stretches along the first principal components of your data, of your training data. And so with this insight, in fact, what happens, what you can show is you can actually use this directly for batch correction. And I think this is quite nicely shown here. So if you use your standard autoencoder, then on this hippocampus data, right, we always have like different mice data. We have mice at 8 months, at 13 months, control disease. So when you do clustering in the latent space, you would want the same cell types to be clustered together, even though they come from different mice or from even control and disease. And you see here that if you, in the latent space, do clustering with these two mice in the standard autoencoder, you see that these cells here are clustered together with all of these cells up here. Right? That's certainly not right. I mean, this anatomical region here has to be clustered with this anatomical region. But all you do is you just use an over-parameterized autoencoder. You don't enforce anything, right? People do all this stuff of like enforcing that the two distributions match between the different mice, between the different batches. You don't do any of that. You just use an over-parameterized autoencoder. And you see here how nicely this works out, right? Where, this cluster here corresponds to this cluster here, and the other mouse corresponds to this one down here and this one. All of this just due to over-parameterization, so no enforcing of anything. And that's really just, from the theory, we do understand why that works. It's just because, it will always work when your biological differences, in this case, the biological differences are much bigger, have a much bigger effect to the overall variation. So biological differences means difference between different cell types, has a much larger effect on the total variation than the differences between the different animals. So whenever you have this, right, at the variation of different cell types, the biological variation is bigger than the variation between different batches, then in fact over-parameterization will just take care of it. Okay, so then you don't need to do any batch correction anymore. This is just going to take care of it. So that's quite nice. Another effect of overparameterization that you actually can prove that something like this will happen. So it directly takes care of all of your batch effects, and leads to these quite nice kind of embeddings here. Okay, so that was just to show at least a little bit of theory of the kinds of things that we're doing when we're actually studying these autoenclosures. So one is to do all these different data integrations, and the other one is actually to then figure out, some theory that can then really help you in terms of actually finding better types of representations of the data. Okay, so with that, I think I should end so that we have some time for actually a different kind of also a discussion about all of this data sets and clinical relevance of it. I just want to end by, of course, thanking my two students, and all of the hard questions that you have will be going to Anit and Shani, and all the other questions will be going to Anthony. And yeah, and with that, thank you guys very much, and let's have some discussion. It's over here.
Yeah.
I will sit here.
There's already some questions, but yeah.
My question about the theory we talked about it, this discrete stochastic system. So how is that kind of clustering as a function of how often you run it through that stochastic?
Oh, so here for batch correction,
yeah,
we actually didn't do any iteration. We just do clustering in the latent space.
I guess like the previous slide.
Yeah, so here, oh, this one.
Yeah, so yeah, the order goes as a discrete dynamic system.
Yep.
So how often do you kind of iterate through that?
That's a very good question. So that depends a lot on your depth. So if you just train on one image and you do a very deep neural network, you won't actually have to iterate. Basically, whatever you put in will just be mapped directly to your training image. So how attractive all of these, so it will depend, for width, somehow what it seems to be, and we're like proving a lot of these results, what width does is somehow making sure that all of your training examples become attractors. So you need to have a certain width in order to do that. And then depth, you can kind of make them more and more attractive.
Is there a relationship between this and score matching models? Because that's also meant to hold how score matching models.
Score matching models.
Score matching models.
We have not thought about that at all. So Adit would be a person to talk to you about that. I'm just going with based on his head that he's saying, so. Yeah, but he would be very happy to chat about.
Yeah, I guess like there's like these score matching models, which like he gives the data and tracks the numbers to it with data points. So it seems that there.
Maybe Anthony can start asking questions.
Sure. So is this on? Okay, good. So first, thanks so much. It's such an honor to have a chance to come and meet with all of you. And so, in kind of thinking about the clinical relevance or medical relevance, a lot of what Caroline talked about, I think it's incredibly high. And I think there are kind of a few different axes that are worth thinking through. One is we're at this kind of interesting moment in time where we're getting very good at human genetics and the ability to discover genotypes that are associated with various diseases. But increasingly, one of the biggest problems we face is going from a variant in the genome to the mechanism of actually how it causes disease. And in fact, if you look over the last something like 12 years, we've gone from knowing less than 10 variants that are associated with common diseases like diabetes or coronary disease to about 500,000. However, for less than 10 of them, do we actually understand the way that they cause the disease? All you can do is point to a place in the genome and say, this increases your risk of getting a disease, but we have no idea how it does that. And so when you think about it, a lot of the work that Carolyn showed, that it was so beautiful, especially as it relates to kind of the single cell biology and the spatial transcriptomics, it really, in my mind, I'm quite optimistic that this will be a very powerful tool to rapidly get at new mechanisms of disease. So that's kind of more from the point of view of drug discovery and basic biology of diseases. And then speaking to some of the later aspects of the work that she presented, I think one of the things that is very clear is that a lot of the assays that we use in the practice of medicine contain a lot of information, especially things like imaging or ECGs or pathology slides. And it's quite embarrassing how little of that information we actually use and how much data we're leaving on the table. You know, you think about, let's say, something like when you do an echocardiogram as a cardiologist, you know, it's so much information. You get not only the shape of the heart at millisecond resolutions over an entire cardiac cycle, but you actually have Doppler, so you know the speed of the blood as it goes through the valves. You get the electrical activity of the heart, also what's going on. There's a tremendous amount of information. And then what doctors do is they take this huge amount of information and they spit out a report that has, you know, tens of bytes of information. And we say the ejection fraction is high or low, and all of this very kind of lossy compression. And what people are starting to realize is actually that using much more sophisticated models, we can actually recover a lot of the information and see things that generations of clinicians have missed. And let me give one example that as a cardiologist, when I first saw it, I didn't believe it. And it blew my mind. And that now actually we're starting to see a lot of examples like this. So a couple years ago, a group at the Mayo showed that you can call a patient's age and gender very accurately by looking at an ECG. No cardiologist I know can do that, you know, or even come close to it. And the fact that, you know, a modern machine learning model can do it so accurately and so well is a really poignant example to me of how much information you're leaving on the table when you let a doctor do the interpretation rather than a machine. And so again, I think that we're just at the cusp of being able to think about how to use this much more in a clinical setting. David, please go ahead. You probably, I hate what I just said. Yeah. Son? Oh.
Yeah. Honestly,
I'll repeat your question.
Okay. Why are you leaving information on the table if you don't know the patient's age? Why don't you ask the patient?
Yeah. So that's a case where
This is, I mean, yeah, I read that paper and I really wondered the clinical significance of it. And I'd love to comment on it.
no clinical significance because you can get the age and gender a lot of other ways. It was more the point that it was possible at all that suggests what other information is hidden in there that we're ignoring. And so, you know, an example that you know very well that's starting to come out now, there's a classic problem of being able to call what patients have cardiac amyloid, right? And that is clinically very important because there's a new set of drugs that are coming out that are very effective for this therapy, and it's very undiagnosed. And so now, what if your ECG machine spit out, this patient likely has cardiac amyloid? That would be really useful. So I think it was more just that the age and gender is suggestive of the possibility rather than the fact that it's clinically useful.
I want to add something to this point. So for instance, both Peter and Adam demonstrated their work that looking at the images of breast mammograms, you can predict weights, you can predict whether women have children, you can predict a variety of things. On the lung, you can predict how much are they smoking. So you can say, why would you go and answer this question? You know, ask somebody. So it is quite well known that for various reasons, people are not saying the full truth. Like, for instance, there are doctors that would not take a patient for surgery if they know that he or she is smoking. And even when we looked at the clinical data, FMGH for mammogram, where there is really no reason to be inaccurate, there were people who wrote different number of children, which was increasing, decreasing, and increasing. There were a lot of numbers, people just very fast write it, and then we build our models on this very imprecise information. So looking at this, and MGH is relatively, educated population, which you would say know how many children they have. It's really all this science built on these questionnaires, we really can't continue this way, and we have to extract from more information. So I very strongly want to agree with you that I think it is important to extract as much as we can.
Thank you. Totally agree. And I guess I'll say, sorry, just one other point I forgot to mention. I was thinking about this beforehand. I talked before about the basic biology and then also the kind of clinical data. one of the things that I think is very important is to develop technologies, both data technologies and experimental technologies, that do more to bridge this gap. And I'll give an example of something that speaks to what might be possible, but again, we're not there yet. A few years ago, there was a paper in Cell from Google Brain where pathologists will often do plain old vanilla staining of cells to look at cancer. And then they'll often do special stains, which are often like fluorescent antibodies that assay, let's say, how strongly is some protein expressed. And again, very dramatic finding that they could actually predict the special stains from just the plain old vanilla, way that pathologists do things. And that just the raw image contained a huge amount of information about the molecular composition of the cells, which again, very surprising to me, having kind of looked at a lot of, pathology slides, the idea that you could actually get molecular information from the gross morphology I felt very exciting. And so, going back to this, if there were technologies that would allow us to take more macro level images of people and be able to gain insights into the molecular understanding of what pathways are active, like why does this person have this type of heart failure? Right now, those don't exist. And you were asking about organoids. That's one really interesting approach to maybe try and build the bridge between the two. But I think being able to cut across these two levels is a really exciting opportunity, but we're not there yet. So sorry, I think there were other questions.
That's a question for Caroline. So I was thinking about these over-parameterized autoencoders, and you mentioned that they can self-regularize. Does that mean they just memorize the training examples, and why like the training examples being a fixed point is a good thing for the model?
Yeah, this is actually a great question.
Can you repeat?
Oh yeah. So the question was about, why is it a good thing that these autoencoders are self-regularizing, meaning that they're memorizing a training example. And so there are many different definitions of memorizing. So usually you wouldn't run this autoencoder this long until whatever you put in gets mapped to one of your training examples, right? So you would do this once, but it is important. You would want definitely a map that is, at least if you're somewhere close to your training examples, remains close to training examples, right? So if you have actually I have it here. So you probably don't want the autoencoder to learn something as crazy as what I have over there, right? That's still an interpolating function that leaves your training example to your training example, but even something that is very close by will be mapped to something far away. So you definitely want it to be, and that's why in fact people have in fact before already developed autoencoders with an additional regularizer that is contractive at the training example. So I think it is a property that you want autoencoders to have. Usually they added a regularizer to do it. Here what we're showing is we'll just over-parameterize and it will do it by itself.
I see. But what if you put another testing image that the autoencoder is not trained on, and we have some random noise, where it's you do something that is sensible, or it's still converted to one of the training examples?
Yeah, but so in fact, when you just have a very shallow network, when your standard way of doing it, right, is you put in your example outcome something, it will look very much to like what you put in. That's why there are even papers that write that the autoencoder learned an identity map, because it looks very much like what you put in. You wouldn't even see that it is contractive at the training examples. It just moves it slightly, and that's, I think, is a good property, right?
Question about the genes and imaging. So you mentioned that there's so much that can be seen from the image, even though there are thousands of genes moving up and down in your body. I think that's because all the genes are moving in tandem, and you're observing the effects of certain pathways rather than individual genes, which can be seen in the image.
It's interesting. I guess let me start by saying I don't know that we know. Now, certainly sets of genes are very correlated with one another, and that's, I guess that's what we believe a pathway is a set of things that go up and down together. And so, it's very likely that there's some kind of subtle changes in the morphology of the cell that correspond to an upregulation of a pathway, and that those together are a stronger effect than the effect of any one gene, and that's why we can predict the expression of the gene. I think that's very likely to be what's going on, but at this point, it's more just an empirical observation, and I don't know that we have a deeper understanding.
So the question related to that is when you do the validation, is it 2 or 3 genes to look at? Were those genes correlated in some or they're random, or like how representative are they of like other types of things? Like maybe those genes are just the same, maybe they don't change that much because like they're so important. Other genes that do change would be have a different outcome.
Yeah, so here actually, so how we chose them were two different things. So one, you need to choose genes where RNA and protein are believed to be highly correlated, right? Because we're predicting RNA, but measuring in the image protein. The other thing is here we looked at T cells. So we actually wanted to be able to predict genes that are relevant for T cell activation, because we wanted to know, like, you know, we actually saw two different classes of DNA packing, one that had more heterochromatin in the interior, one that had more heterochromatin on the DNA in the outside. And we wanted to, for these two different states, we believed that this was related to T cell activation, because also in expression, we saw two clusters, and we wanted to validate that. So we just chose two genes that were very different. One was, you know, more in the active state, and the other one was more relevant for the naive state. And we predicted that this is in fact correct.
But like, does that, like, one of those genes, is it like, if you measure it across different samples or like different cells or whatever the relevant population is, does that change, or is that usually also activated at the same level?
So here we saw two different clusters. We chose genes that were up in one cluster and down in the other cluster, and we predicted that, and that was in fact correct. So of course there is variability, but there were these two clusters where you actually saw two different states in heterochromatin, and they were also two different states in expression, and we were able to predict that. So we chose genes that are functionally relevant because they actually, I mean, functional, they're at least correlated with different states.
Yeah, so I have another question about so the training examples that stored as fixed points. Are there also other fixed points that sort of develop? Like, I'm wondering if you could use this as a generative model where you don't just spit out training examples, but also sort of view images.
Yeah, so no, if you're like super over parameterized, no. If you are in fact under parameterized, then you can go off and you will converge to some random stuff as well. It often looks like something like a mixture of your training examples, but we can actually not prove what it is doing in that setting.
Okay, but you haven't observed it like converging to the image that was
in the under parameterized setting, yes.
Oh, okay.
Over parameterized.
So
Yeah, over parameterized, I think we can now prove this. So until in this paper that I have the reference there, we don't have a proof that there are no other fixed points. I think we now have a proof. It will be written up.
Unfortunately, Adit has a lot of papers to write up.
But he will be most happy to tell you how the proof works.
I have a related question to the over parameterized. Do you mean only like the bottleneck is like very wide, or that it also for
Very deep.
Very deep and broader?
Yeah, very good question. So here, how you can also define it is just by saying that you want some map that can get to zero training error. That's a maybe nice way of saying what we mean by over parameterized. So you are at zero training error, right? And so then the question is like, what is actually, so there are many maps, but this means that they're interpolating solutions, right? So there are many maps that they're interpolating. The question is, what are these interpolating maps actually learning?
So I have a question for Anthony. So when you say, when we're discussing the clinical relevance of something, you often say, we're not there yet, there's more work that needs to be done. Approximately when? Basically, it's like replace MRIs with ECG.
Sure. Good question. So I don't have an answer, not surprisingly. I guess on this one, there's a quote from Bill Gates that I always sort of believe in, which is, we tend to overestimate rate of progress in the short term and underestimate it in the long term. And so I think we're at a moment in time where it's easy to believe that we're going to be using advanced data technologies in the care of patients in the next 5 years. And I think, unfortunately, that's probably too optimistic. And not because the technology won't be there, but the amount that has to change in how we deliver care and the regulatory and the reimbursement landscape and all these things. It's a big change. And I guess I'll give the example that I lived through. I started med school in 2001, which was the year that the genome was sequenced. And I kind of expected by the time that I'm as old as I am now, 45, that everyone would have their genome sequenced and would be using it in the care of patients. And the reality is, even as one of the more genomically informed doctors, around, the number of patients for whom, if I knew your genome, I could actually impact your care is very, very small. It's just a very small subset of things. At the same time, I think if we project forward 20 or 30 years, I think we'll see that the impact of this on the care of patients and public health is, an advance that's akin to at least the discovery of anesthesia or, sterile surgery or antibiotics. I think it is that kind of medical impact that this work will have. It just will take a while.
So thank you very much. Thank you, Caroline and Anthony, for excellent discussion. And I'm sure they can stay for a few minutes if you have questions. Thank you very much.